If you trace the path of fluid deep into the microscopic structures of the human kidney, you move past the porous glomerular filter and dive directly into the complex tubular network of the loop of Henle. The most abundant protein found in healthy human urine is completely absent from the fluid that initially passes through that glomerular filter. This molecule is uromodulin, originally discovered as the TAM horsefall protein. It is manufactured exclusively by the epithelial cells lining the thick ascending limb, or TAL, and the early distal convoluted tubule. The TAL is the specific segment of the kidney nephron responsible for the active reabsorption of sodium and chloride. Standard clinical renal function tests rely almost entirely on measuring glomerular filtration. By design, these tests overlook the metabolic health and structural integrity of the tubular segments. The tubulointerstitium represents the vast physical majority of the kidney's overall mass. In contrast, the glomeruli occupy only a tiny fraction of that functional space. Because uromodulin bypasses the glomerulus, its concentration serves as a direct, quantifiable proxy for the structural health and total intact mass of the kidney's tubules. Inside the cell, uromodulin is synthesized in the endoplasmic reticulum, processed through the Golgi apparatus, and routed directly to the apical membrane facing the urinary lumen. Here, the protein is physically tethered to the outer cell membrane using a GPI anchor. A GPI anchor is a glycolipid structure that secures proteins to the external surface of a cell's plasma membrane. To release the protein into the urine, a transmembrane protease called hepsin cleaves this anchor. A protease is a specific type of enzyme that initiates the breakdown of proteins by cleaving peptide bonds. This cut exposes hydrophobic patches on the protein, triggering a rapid biochemical reaction. The individual molecules cross-link into a massive three-dimensional polymer net that fills the tubular space. This net acts as a specialized immune trap. It binds directly to the type 1 fimbriae of uropathogenic E. coli. Type 1 fimbriae are hair-like appendages on the surface of bacteria that allow them to attach to host tissues. Caught in this matrix, the bacteria aggregate together. They lose the ability to adhere to the urinary tract lining and are flushed out of the body during micturition. Micturition is the physiological process of urinating. This polymer net also physically isolates calcium oxalate crystals, preventing them from aggregating into larger, solid masses. By maintaining these crystals in a dispersed state, uromodulin serves as the kidney's primary defense against nephrolithiasis. Nephrolithiasis is the medical term for the formation of kidney stones. The specific cleavage of the protein and its resulting physical architecture transform uromodulin into a mechanical barrier required for the survival of the renal system. If we shift our focus 180 degrees to the basolateral side of the cell, facing the kidney's interior and the blood, we see a different mechanism. While 90% of uromodulin enters the urine, a crucial 10% fraction is secreted basolaterally into the interstitium and the vasa recta. The vasa recta is the network of small blood vessels that supplies oxygen to the inner regions of the kidney. This basolateral fraction does not undergo the same cleavage, meaning it does not polymerize. This allows the individual protein molecules to circulate freely throughout the bloodstream. In the circulatory system, uromodulin acts as an active immunomodulator, downregulating pro-inflammatory cytokines like CXCL2 and IL-23. Cytokines are small signaling proteins that regulate the body's immune and inflammatory responses. Within the renal tissue, this interstitial uromodulin specifically protects the adjacent, vulnerable proximal S3 tubular segments during periods of ischemic stress or acute injury. Uromodulin is a dual-compartment molecule. It is a localized mechanical trap in the urine and a vital signaling particle required for systemic homeostasis. The UMOD gene presents a unique genetic vulnerability. Pathology emerges at two exact opposite extremes, the misfolding of the protein and its massive overproduction. One extreme is autosomal dominant tubulointerstitial kidney disease, or ADTKD. ADTKD is a rare genetic condition characterized by a progressive loss of kidney function due to damage in the tubular and interstitial compartments. Genetic mutations in these patients disrupt the protein's internal disulfide bonds, causing the uromodulin to become trapped inside the endoplasmic reticulum. This triggers severe cellular stress, ultimately driving the cell into apoptosis, or programmed cell death. These patients present with progressive chronic kidney disease, hyperuricemia, and gout. Hyperuricemia is a condition characterized by abnormally high levels of uric acid in the blood. The opposite extreme involves common variants in the U-MOD promoter region. These genetic variations cause the cell to manufacture massive quantities of structurally normal uromodulin. This chronic overabundance overactivates the NKCC2 transporters. NKCC2 is a membrane protein that actively moves sodium, potassium, and chloride ions from the urine back into the bloodstream. Forcing these transporters to operate at high capacity drives the body to retain excess salt, directly inducing salt-sensitive hypertension. Over decades, this sustained high blood pressure inflicts secondary mechanical damage on the kidney's architecture. Because uromodulin is central to renal homeostasis, any significant deviation from its precise baseline, whether a toxic cellular shortage or an extracellular flood, inevitably drives the kidney toward failure. Current clinical practice relies heavily on serum creatinine to calculate the eGFR. eGFR, or estimated glomerular filtration rate, is the standard metric used to determine how well the kidneys are filtering waste from the blood. The mechanical flaw in this reliance is that creatinine is an inherently lagging indicator. It only signals a problem after significant filtration capacity has already been lost. This chart shows the progression of kidney disease over time. The x-axis represents the advancement of the disease, while the y-axis tracks functional markers. The blue line illustrates the steady decline of functional nephron mass. Notice the red line representing serum creatinine. Even as half of the kidney's total nephron mass dies off, the creatinine level remains perfectly flat. It only spikes upward at the very end of the disease progression. By the time that creatinine spike alerts a clinician, the kidney has already sustained irreversible tubulo-interstitial fibrosis. Tubulo-interstitial fibrosis is the permanent scarring of the kidney's tubular and interstitial tissues. Now look at the green line representing uromodulin data. It tracks precisely with the actual loss of nephron mass from the very beginning of the disease. Because uromodulin is exclusively produced by metabolically active, structurally intact tubular cells, its concentration in the blood and urine drops in exact proportion to tubular cell death. Clinicians can use this proportional drop to detect subclinical AKI following major surgeries. AKI, or acute kidney injury, is a sudden episode of kidney failure or kidney damage that happens within a few hours or a few days. In the context of kidney transplants, falling serum uromodulin levels predict early graft dysfunction and rejection faster than any current filtration marker. Shifting the diagnostic focus from glomerular filtration to tubulo-interstitial secretion eliminates a massive clinical blind spot. Tracking uromodulin provides a real-time, quantifiable measure of the forgotten 90%